everyone, this is 101 Class Survival Guide and today's lecture is going to be about pyrosequencing. The principle of pyrosequencing or sequencing by synthesis was first described in 1993 by Bertil Persson, Matthias Ullin, and Paul Neher. The components for pyrosequencing include ssDNA template, TNTPs, luciferin, DNA polymerase, ADP sulfurylase, luciferase, apyrase. All these are enzymes, these four. This being the substrate, and their functions and their usage will be described. The principle is simple. Just like in the chain termination or Sanger sequencing method, in pyrosequencing, ssDNA template is required as the starting material. As a new strand is being made, the order in which the TNTPs are incorporated is detected, so the sequence can be read as the reaction proceeds, giving it the name sequencing by synthesis. The TNTPs are detected as they are added. The steps for pyrosequencing are simple. The reaction mixture consists of the DNA template, which is the single-stranded or ssDNA template, DNA polymerase, luciferin, ADP sulfurylase, luciferase, and sulfurylase. Luciferin being the substrate for luciferase enzyme. When the correct TNTP gets attached, the signal is generated by a series of chemical reactions due to these enzymes, and which is noted as a light signal noted by the detector and saved on an attached computer. When the TNTP is not attached, for example a non-complementary TNTP appears, then it gets degraded and no signal is produced. The results are generated and detected continually as the strand is generated in the form of light signals upon attachment of the correct TNTP. Alright, let's get into the reactions for pyrosequencing. First of all, we have DNA polymerase that adds DNTPs to the elongating DNA strand. When a correct and complementary DNTP is attached, we get an inorganic phosphate, which is converted to ADP by ADP sulfurylase. ADP in the presence of luciferase as a substrate and in the presence of luciferase enzyme produces a light signal, which is the indication for the attachment of correct DNTP. But what if the DNTP is non-complementary? In that case, apyrase converts it into a monophosphate form. So here we have the reactions. First of all, the couple reactions which are responsible for the production of the light signal. The light signal indicates that a correct and complementary DNTP has been attached. The DNA strand, which has n number of nucleotides being attached, a new DNTP, and if it is complementary, it will get attached using DNA polymerase, and 1 plus means that a new polymerase added DNTP has been attached, and we get an inorganic phosphate here. The inorganic phosphate in the presence of APS is converted into ATP by using sulfurylase enzyme. The ADP with oxygen and luciferin converts into AMP, inorganic phosphate, CO2, oxyluciferase, and light signal in the presence of luciferase enzyme. The light signal is detected by the detector, and it is noted as a signal or a peak depending upon its intensity. But in the case that the DNTP is non-adjacent, or it is non-complementary to the elongating DNA strand, then it gets converted into, first of all, diphosphate form by apyrase and then into monophosphate form, so that it gets degraded and no light signal is produced. Alright, that's it for pyrosequencing. I hope that this lecture was of help for you. Thank you for watching, and please subscribe for more and comment below for any queries or any future requests.